So students, all that traditional biotechnology which we talk about in our kitchen, which are going on in industry, where microorganisms are simply used to give us products of daily use, and that also by only extracting their natural capabilities, is called as the old or traditional biotechnology. Even when we talk about test tube baby formation, which we have covered in the chapter number 4 of the NCERT book, test tube baby formation, all these concepts are also a part of biotechnology. So European Federation of Biotechnology has given a definition of biotechnology that encompasses both the traditional view as well as the modern molecular. Remember, genes or DNA is a molecule, biomolecule. So the genetic engineering is also called as the molecular biotechnology. So European Federation of Biotechnology has given a definition. The definition I quote from the NCERT is the integration of natural science and organism, cells, parts of the organism or parts of the cells, as well as their molecules present in them. Molecular analog is mentioned in NCERT. That means the molecules which are present in them for products as well as for services is called as biotechnology. So when we integrate two things, the two things are the natural science, the knowledge that we have regarding the modification, how can we manipulate, all this type of the knowledge we have is the natural science. So when natural science and organism, now the organism can be a single cell, organism can be a multi-celled, or the organism's portion, that means maybe a simple organelle or maybe the molecule found in that organelle. When such type of things, their knowledge and then how to manipulate them, when these two things join together and give us the products and give us the services, it is simply defined by European Federation of Biotechnology as biotechnology. Now the main thing is, which I was talking about, that the biotechnology is of two types, old as well as the modern. Now modern biotechnology came into existence because plasmids became clear to scientists, and then Stanley Cohen was experimenting frequently over how to remove plasmid from one bacterium and transfer into the another bacterium. And then restriction enzymes, how are they important? All these things became clear. Remember, restriction enzymes were for the first time identified by Arber, but there were scientists along with him also which performed a large number of experiments for successful understanding of the role of restriction enzyme. So Arber, Nathan and Smith, they all got Nobel Prize for this discovery. And then remember, restriction enzymes are also called as molecular scissor, biological scissor, because they cut the DNA at a particular location, reading a particular structure and cut there only. Then there was another enzyme which is called as DNA ligase enzyme. Now in your 11th class, in the biomolecules chapter, last pages, you have learned about the classification of enzyme. So if you remember, synthase or synthetase enzyme or ligase enzyme, when two small pieces of a biomolecule are joined with the help, we always take the help of ligase enzyme, because small molecules are joined to form a larger one. It's just sticking them together, not modifying them. So when two pieces of DNA are there, imagine there is a piece of DNA and you want to insert another DNA into it. So that is only possible, first you have to cut the DNA and then insert. So cutting is done with the help of molecular scissor, and joining is done with the help of the molecular glue. Molecular glue is the other name for DNA ligase enzyme. Now the next concept, the modern biotechnology or the genetic engineering or the molecular engineering is something which is important for us because it has overcome nicely all those problems which we face in the plant breeding and the animal breeding. So when you read the next page of the NCERT, it talks about what are the shortcomings of the plant breeding and the animal breeding concepts. So let me explain to you. So the main thing is plant breeding and animal breeding. Whenever we perform such type of breeding experiment, we are simply exploiting the knowledge of sexual reproduction. So we all know variations can only come with the help of sexual reproduction. Asexual reproduction never give us the variation. So the sexual reproduction provides opportunity for variation and to form unique combination of genetic setup, some of which could be beneficial to organism as well as for the population. Asexual reproduction preserves the genetic information while sexual reproduction permits the variation. Now, traditional hybridization procedures used in plant breeding and animal breeding very often lead to inclusion and multiplication of undesirable genes. See, when we are transferring the sperm for artificial insemination in the animal breeding, or when we are transferring the pollen grain for the plant breeding experiments to be successful, remember we are paying attention to one character which we are interested in. But apart from that one character, the pollen grain or the sperm will carry a large number of other characters also, and some of those characters could be undesirable also. So when we talk about plant breeding or animal breeding or the concept of hybridization, we are handling desirable genes along with the undesirable genes, because you cannot control this. Pollen grain will not carry just your gene, it will carry the complete genome. So when you are transferring the pollen grain or you are transferring the sperm, you are transferring the complete genome, so which is a mixture of both your desired genes as well as the undesired